Good afternoon, and welcome to our weekly Research in Action series brought to you by the Division of Research at Florida Atlantic University. My name is Gina, and I will be today's host and moderator. It is my great pleasure to introduce today's speaker. Dr. Adele Stewart is a research assistant professor in the Department of Biomedical Science at FAU's Charles Schmidt College of Medicine. She is also an investigator in the FAU Stiles-Nicolson Brain Institute. Dr. Stewart received her Ph.D. in pharmacology from the University of Iowa. She is actively engaged in research on FAU's Jupiter campus, and her work seeks to understand how sex-biased brain architecture shapes risk for mental illness using the laboratory mouse as a model organism. Dr. Stewart, welcome. The floor is yours.
Thank you so much for that introduction. I'm very happy to be here today to share with you all a little bit about the exciting research that's going on in our laboratory. So let me just share my screen really quick and we'll dive into the presentation. So as Jean mentioned, the focus of my talk for today is going to be on the role of sex as a determinant of our behavior. Specifically, the importance of sex in determining our risk for neuropsychiatric diseases or mental illnesses. The reason we're so interested in these disorders is, I think, best illustrated by these data. Just to orient you, this graph is showing you stratified by age, the most commonly prescribed medications in each of these different age groups. We have children, adolescents, and teens, adults, and then older adults over 60 years of age. It will probably come as no surprise to anyone that in the elderly in particular, the most commonly prescribed drugs are drugs used to treat things like cardiovascular diseases and to lower cholesterol or treat diabetes. But if we look at younger age groups, you'll find that in fact, the most commonly prescribed medications are those that are designed to treat psychiatric conditions. For example, in children and adolescents, the most commonly prescribed drugs are the CNS stimulants, such as Adderall or amphetamine and methylphenidate or Ritalin, that are used to treat attention deficit hyperactivity disorder, amongst others. If we move into middle-aged or adult-aged people, the most commonly prescribed drugs are antidepressants. Again, drugs that are used to treat depression. So I think that this slide nicely highlights that psychiatric illnesses in particular are not necessarily a large source of mortality, but are a major source of morbidity. That is, loss of productive working hours worldwide. And the costs associated with treating these illnesses are astronomical. My interest in studying the mechanisms underlying mental illness actually began when I was a high schooler, and I thought I would give you a little bit of insight into where my journey in studying neurodevelopmental disorders in particular began. I had a series of odd jobs as a high schooler and college student, ranging from curating the insect collection at the Illinois Natural History Survey to coordinating summer registration for engineering students. But one of the jobs that was both challenging and rewarding simultaneously were the 3 summers that I worked as a teaching assistant in a special education classroom in the rural town that I grew up with in northern Illinois. As a part of this job, I was responsible for aiding in day-to-day classroom operations, and the children that were in this class were those who were going into kindergarten, but believed to need the most support in order to potentially transition them into a typical education setting or continue them on into special education. So this was a summer school class. And again, here we have the most severe children that are in need of the most support in terms of education. And when I was performing my duties in this job, there were a few interesting observations that I made that have stuck with me in the years since. Often behaviors of children that had the same diagnosis of mental illness were very different. And often the therapeutic approach for these children, which at this stage is often aimed at trying to transition them into a typical education classroom, were always best if tailored to the individual needs of a specific child. So this great deal of a high degree of heterogeneity was very typical of these children, who often presented with one of several neurodevelopmental disorders, which might include autism spectrum disorder, intellectual disabilities, other sensory motor developmental issues, or attention deficit hyperactivity disorder. I also noted, when I was working this summer job, that over 2/3, and in some classes, 80 to 90% of the children in my class were male. We had very few girls that were placed in this summer program. And if you compared the behaviors exhibited by males and females with the same diagnosis, those behaviors tended to be quite distinct and often segregated depending on the sex of the child. And I thought I would tell you two little vignettes to illustrate what I mean, talking about two of the children that I worked with very closely when I had this job many, many years ago. One of the children, whose name was Joel, had a diagnosis of autism spectrum disorder. Joel would make no eye contact and avoided all social interaction, a characteristic feature of this particular disorder. He was hyperactive and extremely impulsive. In response to any changes in his routine or sensory stimuli that he considered to be overwhelming, Joel's first reaction was to run away and immediately start pulling everything and anything he could get his hands off of the surface it was on and onto the floor. Obviously, in a classroom, this type of disruptive behavior was often challenging, and getting Joel to sit down and focus on a particular task was a real challenge for us in the classroom. And in fact, these extremely rigid behaviors could only be counteracted in response to a highly preferred reward, which for Joel were graham crackers in the shape of caterpillars. And for those graham crackers, he would be willing and able to sit at a table and focus on a particular task. When describing his behavior at home, his parents referred to him as the Tasmanian devil. On the other hand, we had Antoinette. She also had a diagnosis of autism spectrum disorder, displayed limited social interaction, and often preferred to play alone. Unlike Joel, she was hyporeactive to many stimuli and often quite inattentive. Changes in her routine often made her very anxious, and she displayed a characteristic behavior of many children with autism of repetitive hand flapping or rocking behavior back and forth. She had a limited repertoire of highly preferred interests, and unlike Joel, her parents described her as having her head in the clouds constantly. So at first glance, the behavior of these children seems very distinct, and yet they both have the same diagnosis in terms of their clinical presentation. And in fact, virtually all neuropsychiatric diseases display some degree of sex bias, either in the age of onset, the overall prevalence of the disorder, or, as is the case for the children I just described to you, the symptomology that's characteristic of that disorder in a particular sex. The focus of today's talk is really going to be on neurodevelopmental disorders, which I'll describe to you in more detail later. But I just wanted to note that we have sort of two extremes of the spectrum, with neurodevelopmental disorders such as autism and attention deficit hyperactivity disorder, or ADHD, showing a predominant male bias. On the other hand, disorders such as anxiety disorders and eating disorders, also major depressive disorder, often show a female leaning bias. And the question we then have to ask ourselves is, why? What are the mechanisms that are differentially determining risk for psychiatric illness that causes this risk to segregate based on sex? Before we move on, I also want to make this set of definitions very clear. When I say sex, I'm referring to two of the main categories, male or female, into which humans and most other living organisms are divided based on genetics. This also includes elements such as the presence or absence of a gene located typically on the Y chromosome that causes the male pattern of reproductive development, also the types of gonads, sex hormones, reproductive anatomy, and external genitalia. So sex refers to the biology of the individual. Gender, on the other hand, is a personal and societal construct, the perception of one's sex based on the behaviors and other expectations that our culture has placed on that sex. Again, gender has sort of inherent biases and predetermined norms of behavior that are typical. So, for example, we may expect women to be more emotional and more in tune with their emotions than a man. That would be something we would categorize as a gender-dependent behavior. Sex, though, again, is all about the biology. There's quite a bit of controversy surrounding the idea of what is actually causing this sex bias in neuropsychiatric disease. And what it really boils down to is a nature versus nurture argument. Is it that males and females are behaviorally different and that these differences have biological underpinnings? Or does biology play no role, and instead our societal expectations for gender-based society will result in gender-biased brains? There's also another component to this, and that has to do with the differences in incidence of psychiatric disease potentially being driven by a failure of our diagnostic criterion and detection methods to actually be able to detect a disorder in one sex or the other. There are proponents and opponents of all of these different arguments on either side of the aisle. But I wanted to highlight one of the opponents of the idea of a male or female brain. If you're interested, you can go and look further into this argument, but Gina Rippon really argues that there is no evidence of biologically driven differences between the male and female brain, and instead it is society that is determining the behavioral differences that we tend to see when comparing men and women. She also points out that research on sex differences has historically been quite problematic, often misinterpreted. There's issues with publication bias, weak statistical power, inadequate controls, etc. These are all valid points, and the expectations society has placed on each gender probably do play a role in determining some aspects of disease risk. Rippon also points out that there's a danger when trying to establish biological or behavioral differences between the sexes, and the use that such science could be put to in order to reinforce male and female roles and status. An effect she refers to as neurosexism. So you can see that there's quite a bit of controversy surrounding these ideas. And the question then is, how do we go about teasing all of this apart? How do we remove from the study of potential sex differences in the brain the potential influences of our gender biased society on these outcomes? Before we dive into the potential utility of model organisms in answering some of these questions, I wanted to show you a little bit of the data that Rippon is referring to when she argues that there are no or few fundamental biological differences between the sexes. One of the sort of critical studies, or a couple of the critical studies, were published a few years ago, and these are looking at structural differences utilizing MRI between the brains of men and women. In this particular study, with the data on the left, what you're looking at is differences in volume, surface area, or cortical thickness in the brains comparing men and women. If one of these is more male leaning, i.e. higher in males, it will show up as blue. If one is more female leaning, it will show up as pink. I think what you can appreciate is if you just look at the average differences in brain volume or brain surface area, male brains tend to be larger. Female brains, on the other hand, tend to have increased cortical thickness. The thing is, though, if you normalize these measures based on the height, weight, or general size of the individual, all of those differences go away. And in fact, these base or apparent differences in brain size are really only a function of the fact that men tend to be larger. The image on the right is showing you two overlapping distributions of average brain volume between males and females. And while the averages tend to be different, what you'll note is that there's a great degree of overlap in brain volume between the two sexes. And there's only a few individuals that are at one extreme end of the distribution or the other. So at first glance, it really looks like there are differences in brain size, but when we look closer, we see that it's really just a function of an individual size and that there's a great degree of overlap for this particular metric. However, there are some more recent studies using newer technology that have looked not at brain structure, but actually the activity and function of the different circuits or patterns of neuronal connection within the brain. In this particular image, what you're looking at is functional MRI, looking at connections and the strength of connections between different critical regions of the brain. The image on the left is just giving you an idea of the fact that many of these connections are pieces of networks in the brain that are important for different behaviors, for our ability to see, our ability to move, our ability to hear, our ability to pay attention and remember, etc. So again, the image on the left is separating these connections and telling you which connections are stronger in females, and those would show up in red, and which connections are stronger in males, those would show up in blue. So what you can see is even though the structure of the brain doesn't differ much between the two sexes, there are quite a few differences in how these regions of the brain are talking to each other. And each of these networks is important for controlling a different behavior. There are also a few additional studies that have looked at general sex differences in cognitive function. I should note, in a study published in 2008, that was performed in Scottish age school children decades ago, overall, there is no difference in intelligence as measured by IQ as a metric between males and females. But there are some more subtle cognitive differences that you can often pick up when testing women and men across a series of different cognitive tasks. For example, in general, and again, this is on average, women tend to be better at reading comprehension, at fine motor coordination, and at retrieving information from long-term memory. Men, on the other hand, tend to be better at visual-spatial type of tasks, as well as at multitasking or performing more than one task at the same time. Interestingly, if you look at a spatial navigation task where an individual is required to navigate from point A to point B, in those tasks, both of the sexes perform the task to the same level and are capable of navigating the task, but they use different information in order to perform that task. Women tend to use landmarks in order to navigate through space, whereas men tend to think about more allo or egocentric information, i.e. the relationship between their self and other pieces or points in space. So if you and your spouse have ever argued or gotten into an argument trying to follow directions or find your way to a particular location, might be because you're actually speaking a slightly different language and need different information about the different landmarks and other spatial information present in order to properly navigate that space. Also in infancy, females tend to react more to faces and males to objects. So there are differences, right? And the question is, why are these differences there? I also want to take a moment to highlight some of the consequences of ignoring sex differences. And the most famous example of this is referred to as Yentl syndrome, so named based on the movie starring Barbra Streisand, in which she dresses up as a boy in order to study at a Jewish synagogue. This syndrome specifically references the underdiagnosis and misdiagnosis of women's cardiovascular disease because of an inherent male-leaning bias in our diagnostic criterion and treatment. So men are actually more likely to experience an ischemic heart attack or to have some sort of cardiovascular medical problem. But women are more likely to die from these disorders. The risk is 2 times more likely for women who are young. And if you're a woman who presents with the symptoms of ischemic heart attack that are more similar to those that men often exhibit, you're more likely to be quickly diagnosed and properly treated. So, as with Barbra Streisand dressing up to look like a man, for ischemic heart disease, if you dress up and look like a man, you're more likely to have a better outcome. In basic science research, we're actually doing a very bad job in general of considering sex as a biological variable. One of the most highly cited studies was performed in 2011, and what this study concluded was that although sex and potentially gender are important contributors and determinants of psychiatric disease risk, most basic science research omits females from their study. In neuroscience and pharmacology, these particular disciplines are actually amongst the most strongly skewed. And in 2011, the ratio of studies exclusively utilizing males versus exclusively utilizing females was almost 6-1. So why? Why aren't we including females in our research? Well, circa 2015, many researchers argued that there were no sex differences in brain function outside of the context of reproductive behaviors. There was also the highly cited concern that hormonal cycles due to menstrual cycling in women or the estrous cycle, as an example, in rodents, that change in hormone levels would decrease the homogeneity of study populations. Really, what this boils down to is that controlling for sex would require more animals, more resources, and more time. And in fact, many of the concerns that have been highlighted against the inclusion of females in these basic research studies are myths. In fact, research has shown that males exhibit equal or greater variability than females due to hormonal fluctuations in testosterone that often result from group housing animals and the establishment of social hierarchies. Analysis of data sets looking at gene expression patterns across male and females actually showed that females are no more variable than males in measures of gene expression in both humans and model organisms such as mice. And finally, in a study that was published in Cell in 2019, a large meta-analysis of proteomics data sets, this is protein expression, actually determined that the variability between individuals was pretty heavily influenced by sex. About 13.5% of the variation of expression could be attributed to the sex of the individuals. This was more, in fact, than other environmental factors such as diet, which is often cited as something that will influence gene and protein expression. So we're excluding females from research, and it was actually in about 2011 that the NIH, which is one of the largest funders of basic science research in the United States, identified this as a problem. In 1993, women were required to be enrolled in clinical trials, but it wasn't until 2015 that the NIH required basic science researchers to include female subjects and make allowances for consideration of sex as a variable for their basic science research. However, the addition of that mandate has yet to make a large impact on our inclusion of female subjects in research. And in fact, when comparing the number of studies utilizing both sexes of animals and considering and separating their results by sex, there's actually been no change in neuroscience in this metric in the decade between 2009 and 2019. A lot of the studies that have been published describing sex differences have focused on descriptive rather than mechanistic elements. And because much of our scientific literature has been derived from the study of males, many of the tests and other experimental paradigms that we've established are also designed and validated in males. And where does this really get us? When we want to take information from the human clinic and use that information to generate models of diseases that will give us new treatments, new diagnostic criterion, new biomarkers for disease. All of these elements are essential to utilize these model organisms in order to study. Well, our lab has been for many years interested in taking genetic information from human patients, identifying rare genetic variants that are potential contributors to disease, putting those variants into model organisms, such as the laboratory mouse, in order to use those organisms to potentially identify new mechanisms of disease, screen for new therapies, and also improve our biomarkers and diagnostic criterion for psychiatric illness. We've been heavily focused on neurodevelopmental disorders such as autism spectrum disorder and ADHD, or attention deficit hyperactivity disorder. And notably, the prevalence of these disorders has been increasing over the last few decades. And as I mentioned earlier, these disorders are much more common in boys than they are in girls. The mutations that we've looked for are relevant to one particular neurotransmitter system in the brain, and that is the dopamine neurotransmitter system. Dopamine is a chemical present in the brain that mediates communication between different sets of neurons. And the neurons or networks that dopamine is important in controlling are important for a number of different behaviors. Many are familiar with dopamine's important role in movement because dopamine producing neurons are the neurons that die in individuals with Parkinson's disease, leading to their issues with motor control and movement. Dopamine is also heavily implicated as a mediator of reward. Pretty much anything that you consider to be pleasurable, whether natural or not, is going to cause an increase of dopamine in the brain. Dopamine is also very important in mediating a number of higher order cognitive functions, including memory and attention, and has been implicated in a number of neuropsychiatric conditions. We focus on one component of this neurotransmitter system, that is the dopamine transporter. I don't need you to understand all of the elements of this particular diagram. We're going to focus on this protein in pink, DAT, and what it does in controlling how dopamine mediates a signal that moves from one neuron to another in the brain. Dopamine is released from one neuron and activates the next neuron. The transporter sits on those dopamine releasing neurons and clears dopamine back into the synapse. It mediates how long and how much dopamine is present to activate the next neuron, and is also an important mediator of recycling of dopamine back into the neuron so it can be released again. Importantly, this protein is the target of a number of very important drugs, including the CNS stimulants I mentioned at the beginning of the talk, methylphenidate or Ritalin, and amphetamine or Adderall, as well as the drug of abuse, cocaine. All of these drugs bind and block the dopamine transporter. We screened patients with ADHD for mutations in the dopamine transporter and identified one single point mutation in the dopamine transporter, which I'm going to refer to as the Val559 mutation for the rest of the talk. This mutation was identified in two brothers with ADHD and was previously identified in a girl with bipolar disorder, and subsequent to it coming to the attention of our lab, found in two boys with autism spectrum disorder. So it appears that this particular mutation might not necessarily increase risk of a specific psychiatric condition, but generally increase risk for several disorders, all of which have some dopaminergic component. What does the Val559 mutation do? Well, not to go into too many specifics, but normally the function of the transporter is to clear dopamine. And by doing so, it shuts down communication via dopamine between those two neurons. The Val559 mutant can clear dopamine and then it just spits it back out. So in the panel on the left here, we're looking at clearance of dopamine using a method called in vivo chronoamperometry. We add dopamine onto slices isolated from the brain. And in purple, you see that initially our signal indicating extracellular dopamine goes up, and over time, the signal disappears. And that's because the transporter is clearing that dopamine from the extracellular space. In orange are our mutant mice. And what you see here is that they can clear dopamine, but it's less efficient. And as a result, here in the right, they exhibit, again, orange is our mutants, an increase in dopamine levels tonically in their brains. What we want to know is what happens when an animal expresses this mutation. And we're particularly interested in what happens to rodent behavior, specifically behaviors that might be relevant to the psychiatric disorders in which the Val559 mutation has been identified. So for autism spectrum disorder, the real big clinical components of that disorder are changes in social behavior and repetitive behavior. For ADHD, we want to know about impulsivity, attention, and activity. And for bipolar disorder, we're more interested in things like anxiety-related behaviors, in reward and psychostimulant sensitivity. We've done a lot of behavioral characterization of these mice, but in the interest of time, I'm going to walk you through a few interesting experiments. First, looking at social behavior. This particular test is called the tube test, and it's a very simple test of social dominance in mice. You take one wild type or normal mouse, and on the other side, a mutant mouse. They're placed on either side of a tube that's just big enough for the mice to be able to walk through the tube, but not big enough so as to allow them to turn around. The animals will interact in the center, and one mouse, the more dominant mouse, will push the other mouse out of the other end of the tube. That mouse would then be declared the winner of a bout. And what we noticed was that our male mutant mice win more bouts here when they were tested as adolescents. The interesting thing, though, was we didn't see any difference in win numbers in our female mutant mice. So we see this sex-specific behavior of our Val559 mutants. We've also looked at a simple test of social interaction. Here, just asking a mouse if they prefer to be close or far away from a mouse that they've never seen before. And again, our males show a reduction in the time they spend interacting with a novel mouse, but not our females. We've also done a few cognitive tests, including a very simple test of memory. Here, on a single first day, the animals are placed in an arena with two identical objects, and they're allowed to explore. Mice are naturally curious, and they'll explore each object and the rest of the space. On the next day, we replace one of their familiar objects with a new object. And generally, wild type or normal mice prefer to explore the novel object because it's new and potentially exciting, whereas the other object they've already explored and it's familiar to them. This test tests two aspects of rodent behavior. One is their interest in something new, and the second is their memory and their ability to recall whether or not an object is new or familiar. Here, unlike what we saw with the tube test, our Val559 females have the issue. Here we're looking at heat maps which are telling you where the rodent explored in this arena. On the novel versus novel day, that's the first day, they're given two identical objects, and all the mice pretty much spend time exploring each object equally. On the second day, when one of the objects is replaced by another novel object, you'll note that the males, regardless of genotype, and the female wild-type mice spend a lot of time near the new object, but the female Val559 mutants don't. So what we have here are behavioral issues in our model that are sex biased. It's not that there are different behaviors or different degrees of deficit in each sex, but in fact, the two different sexes of mice exhibit completely different behavioral phenotypes. I'm going to skip ahead a little bit in the interest of time because I want to make sure that we have enough time for questions, but we've performed additional tasks, and I can tell you, after performing a number of different assays on our mice, that this theme holds true. That is, in most cases, we have behaviors or changes in responses to drugs or in baseline behaviors that are the same or that are different in each sex. Some of these behaviors are altered only in one sex or the other. And in some cases, as with the light-dark transition test, the phenotype that we observe, the behavior that we observe, is in one direction in the males and the opposite direction in the females. The question that we're really working on as we move forward on this project is, how is this possible? These are mice, right? They all have the same mutation. They're not living in a gender-biased society as far as we can tell. So what is it about the environment that's present in the brain in each of the different sexes that is causing the same mutation to have completely different behavioral outcomes? And we've identified a few mechanisms that might be important. One of them has to do with another piece of the system that's present on these dopamine releasing cells. It's called the D2 or dopamine 2 receptor. And this receptor, you can think of it as a sort of negative feedback loop on the system. Dopamine is released, it feeds back onto this receptor to shut down further dopamine release. And in fact, what we found is for the behavioral phenotypes, such as this deficit in the novel object recognition task that we observe in our Val559 females, if you block that dopamine D2 receptor with the drug sulpiride, this drug is used clinically as an atypical antipsychotic, you can actually restore the performance of our Val559 females in this task. So recall again on the second day that we're looking for the animals to prefer to spend time with the novel object. The wild types do, and sulpiride doesn't really impact their preference. The Val559 mutants, if you just give them saline, still fail to spend a significant amount of time with the novel object. But now, if you give them sulpiride, they spend more time and prefer the novel object. In another task that I didn't have much time to explain, but that exhibits a male bias phenotype, this spontaneous alternation test of working memory, we see the same thing, that with this drug that's targeting the same receptor in each case, we can normalize the behavior of our Val559 mutants. What does this all mean? Well, we're sort of working with this framework for our studies moving forward. And it's this idea that there are fundamental differences in the architecture of the dopamine system. For example, how the D2 receptor is modulating dopamine release and reuptake, that are fundamentally different between the two sexes. And as a result, if you take a single perturbation of the system, such as our genetic variant, the VAL559 mutation, this sex bias architecture will send the animals in a different trajectory of behaviors. So we currently define our mental illnesses based on behavior, but in fact, it might be possible that disorders we define as categorically different based on behavior have the same biological underpinnings. We've identified a few mechanisms ourselves in this lab, but we're not alone in trying to understand all of the potential differences that exist in this particular system between the two sexes, of which there are many, and probably many more yet to be discovered. There are also a number of risk factors for psychiatric illness, including other genetic variants, epigenetic influences, environmental factors, experiences such as stress, trauma, nutrition, exercise, etc., exposure to drugs, either prescription or illicit, that will also be filtered through this sex bias architecture and may thus differentially impact males versus females. And we think that these mechanisms, this sex bias architecture, is probably a very important determinant of the sex bias that we observe for many psychiatric illnesses. And it should serve, I think, as an impetus for us to begin moving away from a behavioral definition of psychiatric disease to one that is biological. A lot of people I have to thank for the work that I've described in this particular talk, including a number of members of my lab, also funding sources from the Brain and Behavior Research Foundation and the NIH. And with that, I would be happy to take any questions you may have about the research that I presented.
Thank you, Dr. Stewart, for what a wonderful presentation that was. Very in-depth, I'm sure, because I'm sure that it's somewhat of a difficult thing to explain this down to that kind of detail. So let me see if maybe some of the questions that I have for you, you might be able to address them in maybe simpler terms. For instance, when you were describing in the slide regarding the social behavioral test as an example, what wasn't noted on there was what would be typical of normal behavior, I guess, in mice versus the mutant. So the differences in also female-female interaction in normal mice, male to male or female-male, since obviously you mentioned before, to our knowledge, mice aren't gendered, but they are biologically distinct.
This is a great question. So in the tube test, if you take normal mice and pit them against each other, you'll end up with a 50-50 split, right? About half of the time, the mice are going to win, half of the time they're going to lose if you separate them into two groups. For wild type mice, this particular test has historically been used to establish dominance hierarchies in group housed animals. So you can utilize the test in a group of mice that are living together for a long period of time in order to figure out who the most dominant mouse is in the group and the least dominant. It does work better in males, but females also establish these dominance hierarchies, and you can utilize the same test in order to establish a dominance hierarchy in females. Importantly, many models that have linked mutations to, in particular, autism spectrum disorder show alterations in their dominance exhibited in this particular task. So it's one of the reasons we utilize the tube test. It's also a very simple test and very amenable to the addition of pharmacological manipulations to try to reverse or rescue behavioral changes we may see in the mice. Most people don't pit one sex against the other in general, but in animals living in the wild, you could do so and would establish a hierarchy. In lab mice, they're housed either all males or all females, so we generally don't do those types of analyses.
I see. There was another question that came in, an anonymous question. I'm not sure how to phrase it. It says, long-term use of medication will change any effects of behavior. Not sure how to reword that question.
Okay. There's a few different layers to this type of question. You could, both in the context of therapeutic use of drugs and in the context of abuse of drugs. So to take a single drug like amphetamine, which you may be more familiar with as the drug Adderall, this is a drug that is used therapeutically in the treatment of ADHD and also abused for various purposes. As it increases dopamine in the brain, it is also an addictive chemical. And clinicians often have to balance that addiction liability when prescribing the drug. Long-term use of amphetamine changes the brain in many ways. And what we're learning is that how it changes the brain differs between the two sexes. As an example, women are actually more susceptible to amphetamine abuse, though males use amphetamine at a higher rate. If women do use amphetamine, they are more likely to become dependent. They're more likely to reach the diagnostic criterion for abuse, and they tend to have a difficult time weaning off of the drug and maintaining abstinence from the drug. This is also true for cocaine, which is another drug that modulates DAT. We are just beginning to delve into the impacts that many of these drugs, not just the ones I just said, have on the brain. And many of the impacts are different if you compare the two sexes. We're at a huge disadvantage, though, because, as I mentioned early on in the talk, we have decades of research done on these drugs that has been performed only in male animals. And we have very little information about what happens when you expose a female to these same drugs.
And that actually leads to my next question regarding the drugs themselves and used in your presentation, sulpiride.
Mm-hmm.
So in drugs like that and similar drugs that have been for psychiatric or neuroscientific kind of disorders, were they all then developed specifically for the male brain or have been used? So it's kind of the reverse engineering that we're going back now and looking at those drugs specifically in the female brain?
Yes. I mean, really just as simple as that. There has been no requirement until 2015 for researchers doing the type of research that goes into developing these drugs. They have never been required to use females and to assess whether or not the impacts of those drugs would be different in females. It has never been a requirement. And as I showed you on the slide where I compared the publications that we're seeing in 2009 versus 2019, even the addition of that NIH mandate hasn't really done enough to force researchers to include females in their studies. I personally sometimes get a little angry when I think about this. And, I, for example, have a friend that I know very closely, and she has struggled for many years with finding an appropriate therapy. She's someone who struggles with depression and post-traumatic stress disorder. And I once sat down with her because she wanted me to explain to her how these drugs work, how they were developed, and why her therapist kept prescribing a bunch of different drugs to her. And when I told her that all of the drugs that she was taking were developed only in men, even though the disorders that they're designed to treat are more prevalent in women, she was very angry. And that sort of, it's something that's very motivating to me, but at the same time is also very daunting. We have a lot of work to do in order to figure out, for all the drugs that are currently on the market, have we sufficiently characterized their impacts, not just in each sex, but in individuals with different genetic or other backgrounds? So there's a ton of work to do, and I wish that I had better news to share with all of you for that type of question.
A lot of work to be done. There is one more question that just came in from a Jenny Lacombe. What about the individual variation in males and females? For example, some men have higher or lower levels of testosterone naturally. Would this variation within one sex impact the expression of the dopamine architecture?
Yes, this is a great question. And it's something that is sort of a next step for us. I mentioned a study that was performed a few years ago where they actually looked at gene expression variability. And in both males and females, obviously, there's inter-individual variability. And it's about the same, actually, in each sex. So one sex is not more variable than the other. But certainly, if you compare within individuals in each sex, there is going to be variability that will result typically, if we think in the laboratory mouse, in females, it will result from estrus cycle fluctuation, so changes in levels of estrogen and progesterone across that estrus cycle. And in males, the variability has been predominantly attributed to differences in testosterone that result from a social hierarchy that is established in group-housed mice. So both of those things likely also contribute. There is some very interesting studies that have shown that estrogen actually directly controls a number of aspects of the dopamine system, including the dopamine transporter itself. So we're very interested in subcategorizing our mice based on estrus stage in order to control for their levels of sex hormones and see if that's something that will influence outcomes in the mice. Similarly, you could do something for the male side of things, looking either at separating the mice based on their social standing, monitoring testosterone, or adding the addition of a gonadectomy, in order to remove the confounding influence of testosterone. So a great point. And again, just sort of highlights how much more work there is to do on this particular topic.
Definitely. Dr. Stewart, thank you very much for your time, and we wish you a great afternoon, everyone.
Thank you. Thank you all for having me.
Thanks so much. Bye-bye.
Bye.